Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Graziella Piras from 908 Devices.
Thank you so much, Leah, and welcome everyone. Good morning, good afternoon from wherever you are. Today we're going to look at the critical role of cell culture media analysis in cell and gene therapy development. We will first look at expert recommendation, a quick overview of the literature and what we can learn from what is already known, the impact of cell culture analysis on cell and gene therapies. We'll look a little bit at the biology of T cells. What tools do we have at hand to analyze cell culture medium, spend media? And then, very importantly, we are going to look at very recent data from two groups. First, from Emory. They're studying CAR T cell activation and correlating with amino acid profiling. And then data from MIT, who are working on viral vector production, optimization, and modeling. So what can we learn specifically for CAR T cell therapy development, how cell culture media really matters? Actually, there's two aspects here that we can find in the literature. One is that especially for CAR T cells that are used to treat solid tumors, these cells have to be metabolically ready to fight the tumor environment, and they have to be cultured ex vivo to be able to do that. And then an example at the bottom there of a report showing that specific amino acids, the T cell activation is really depending on specific amino acids. So yes, cell culture medium matters, and we have to optimize it, and we have to have ways to look at it. So if we focus on the T cell activation, the ex vivo portion of CAR T cell manufacturing, we can find tools to metabolically prepare these cells and activate them. We learned that formulating cell culture media and specifically key nutrient composition is very important for their activation, expansion, and differentiation. An interesting quote from this review reads that actually commercially available cell culture medium that is designed for T cells may actually be not appropriately balanced to ensure CAR T cells are effective in vivo. So there's work to do to optimize these media, and we'll see some data about that. So what are the tools that we have at hand to measure the cellular and the extracellular environment for these cells? Well, very well-known methods or very well-established methods are available to measure viability and viable cell density and cell size, key metabolites such as lactate and ammonia and others, and of course, physical parameters. And these tools are available at the point of need, usually at line or online. What is, however, not available, usually at line right next to the bioreactor, are methods to measure amino acids and vitamins, which are key nutrients in cell culture medium. At 908 Devices, we provide a cell media analyzer that specifically allows for measure of amino acid at the point of need. This instrument is small, very easy to use, and allows measurement of these key nutrients so that we can correlate and optimize them to prepare the cells, for example, in the case of CAR T cells. So we've learned that CAR T cells need to be cultured ex vivo in the appropriate cell culture medium for proper activation, proliferation, so that they're also metabolically ready to fight the tumor environment and have anti-tumor response in vivo. This requires frequent at-line measurement of key nutrients such as amino acid, and the REBEL enables such medium optimizations. What can we learn from T cell biology? What we learn is that several key amino acids are required for T cell activation and proliferation. And we have some examples shown on the slide here. Leucine and glutamine and arginine and alanine, many others, have key roles in T cell differentiation, activation, but also to provide that very important anti-tumor immunity. So what we did, if you remember, one of the observations in one of the papers was that commercial media may not be well balanced to allow CAR T cells to have good tumor response. So we actually looked at five different media that are designed for T cells available off the shelf from media vendors, and looked with the Rebel at their amino acid profile. And we can see here, if you remember from the previous slide, T cell biology tells us that key amino acids are required for T cell activation and anti-tumor effect. And you can see that some of these amino acids we had on the previous slide, like alanine and arginine and leucine, vary widely, and arginine vary widely in these different media. So you would want to know what's in your media to start with, and then maybe how to supplement the right amino acids that are key for your process. So I am very excited to be able to present to you today two case studies. The first one is a case study that comes to us from Emory University, from the laboratory of Eric Dreden. And his team are looking at novel CAR T activation process and correlation between CAR T activation and metabolite profiling. So initial CAR T response is very promising. However, it has been shown that long-term anti-tumor effect, durability of the anti-tumor effect is not always there. And it has been shown in the literature that specific CAR T cell phenotype can correlate with durability of the effect in vivo. And we are talking about here about naive, less differentiated T cells correlate with durability of anti-tumor effect. So the team in Dr. Dreden lab are developing microbead libraries that are used for activation of T cells. And these microbeads are conjugated with several immunomodulating proteins such as IL-2, IL-15, and anti-CD3 and 28 in various amounts. So they've generated a library of compounds, and they're testing them to see if they can actually increase the proportion of these naive cells. And indeed they do. If we look at the graph at the bottom here, at the bottom in the middle of the slide, we see that some of their compounds, numbered 1 through 50, actually provide equivalent or higher number of these naive T cells compared to the industry gold standard, which is Dynabeads and IL-2. In addition, what they're doing is establishing structure metabolism relationship to identify if these desired phenotype of naive T cells correlate with specific metabolite profile, and they're using the Rebel to do this. So in the experiment shown here, CAR T cells were manufacturing using a 50-compound microparticle library, and then the supernatant post-manufacturing was analyzed with the Rebel. And if we look at the graph on the upper left here, we're looking at 3 amino acid and 1 dipeptide in a few of their compounds, and the comparison with the industry standard Dynabeads with IL-2 or IL-15. And what we observe, actually what they observed, was that concentration of alanine and glutamic acid are lower in some of their microbead compared to the Dynabeads and IL-2. And you can see this clearly on the graph on the bottom here, which shows the fold change of amino acid over this, the commercial standard. So this is very encouraging. They can start to see some specific patterns linked with that desired phenotype. And their compound 16 and 27 were among the ones that had that high, naive, less differentiated phenotype. So in their next steps, they're working to further confirm these profiles and establishing the correlation between activated T cells with this phenotype and nutrient intake. The goal is to eventually being able to prolong this anti-tumor effect of the activated CAR T cells. So I'm going to switch now and talk a little bit about gene therapy and viral vector production. So what does the literature tell us in terms of how to increase viral vector titers and quality? I just picked up one review, but there's a lot of literature around this showing that actually the metabolism of HEC293 cells, which are frequently used to produce viral vector, influence the production, the level of production and the quality also of these viral vectors. And specifically, we can see some quotes here that say that, for example, for adenovirus production, asparagine is an essential amino acid for formation of variants. Another interesting fact we can pick up from media vendors is that the medium used to produce viral vectors, specifically in this case in HEC293 cells, really vary widely with media composition. And they recommend to actually further optimize the medium because every cell line, every system is different. So there is a need for medium optimization. We picked up 5 different HEC293 media here, and we see, for example, that asparagine, which is known to be important for adenovirus production, varies widely. So same as what we saw with T cells, you want to know what's in your medium so that you can further optimize it. So I'm going to now switch and go to the case study we have on viral vector production and talk a little bit about the work that's being done at MIT on viral vector production. So typically viral vector AAV, recombinant AAV are produced with using triple transfection in HEC293 cells. This works pretty well. It's pretty well established. However, viral titers are still very low and way too low to be able to use these therapies with actual indications. Also the capsid quality, so there's a lot of empty capsids, and this needs to be remedied as well. So the team, and there is also very little mechanistic understanding of how metabolite consumption can guide the development of more advanced processes to overcome these challenges. So the team at MIT are actually developing a model to design and predict capsid formation to achieve high productivity and high capsid quality. So they published initial data last year, shown here on the left, and their model has been verified in shake flask, as we can see a good model fitting for capsid and AVDNA. And they will use this model then to guide development of continuous bioreactor AAV production. So a series of next experiments are being done. One shown here, a fed batch in 300 ml microbioreactor is being done to assess depletion of amino acids using the REBEL. So twice a day, samples are taken and analyzed on the REBEL. And what they have observed, as shown on the right here, is interestingly, 2 of the amino acids that are known already to exhibit high flux of consumption in HEC293 for AV production showed indeed high depletion in their experiment, but also some other amino acid. So they will use this data and further examination of the process, basically, and using the REBEL to design better feeding strategies, but also inform development of continuous process at bioreactor scale. So can we leverage cell culture medium analysis and optimization for cell and gene therapy? I believe yes. And I've shown you 2 great examples where the group at Emory and MIT are doing exactly that, identifying key metabolites to further the development of these therapies. You can learn more about this in an ebook that is on our website. I encourage you to go download it. And I would like to thank you very much for your attention. Back to you, Leah.
Okay, great. So before we get started with the Q&A, we do have some questions here for you in our audience. So on the screen, if you can go ahead and just select your answer from the questions that you see. So do you currently monitor amino acids in your process? No, I have not considered it so far. No, but I'm interested in doing it. Yes, but infrequently. yes, frequently using an external lab, or yes, frequently using our in-house lab. So that is your audience question. And then now we are ready to go to the Q&A. And we have our first question there. So how often do you recommend measuring amino acids in the CAR-T process, Graziella?
Yeah, thank you for the question. So in the work at Emory University in Dr. Dreden lab, they at the moment they measure amino acid just at the end. But of course, the more measurement you do throughout the process, so you can see sort of the evolution of the amino acid consumption, the more information you will get and the more input you can have on maybe understanding if some amino acids are being depleted or some overproduced. So the more information throughout the process you can have, the better.
And what is the most important amino acid that needs to be optimized for adenovirus production in the HEK293 cell line?
So I believe we had an example here with, I believe it was from the MIT team. We saw 2 examples of 2 amino acids that are very important. So I can go back to that. And it's been demonstrated already. Asparagine and aspartic acid are known to have high flux, so they're highly consumed by the cells. But I think every system can be a little bit different, but these 2 definitely are very important. But we can see from the experiment that there are some essential amino acids being depleted. So these must be also highly consumed. And so I think basing your experimental design on known information, but also observing your own system to see what is needed always.
And what is the workflow for sample analysis?
Okay, so I believe on the Rebel is the question, yes. So actually the sample preparation is very simple. All you do is take your samples out of your bioreactor, spin out the cells, and it's ready to go to load on the Rebel, where you will dilute it with the Rebel diluent, and the instrument does the work for you. So there is no protein removal, nothing like that, just removing the cells, and it's ready to go on the Rebel.
Okay. So as a reminder, you can type in your questions. And if we don't get your question today, we will be passing those on to Graziella and she can follow up with you directly. I was going to go ahead and make sure that your contact information is there on the screen. Okay. So, well, thank you, Graziella. It looks like that's all the questions we have for this webcast. So thanks to you and thanks to our audience for joining us. The recording of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.